It is my great pleasure to introduce to you Becky Hardman. Some of you, maybe most of you know Becky. I've known her for many years now, and I know that makes us sound really old. Well, I'm really, really old. She's not so old, but she's an awesome person. She's done amazing things, and she was here at UT for a while. But she's a wildlife veterinarian now with the Florida Fish and Wildlife Commission. She received a BA in Ecology and Conservation Biology from Boston University and an MS in biology focused on herp ecology at Western Carolina University. And that's really when I first started getting to know her. Then she moved to University of Tennessee, where she actually started working as a lab tech. But then she went to vet school and she earned her DVM and her PhD focused on wildlife health. She continued at the University of Tennessee as a postdoctoral researcher for the One Health Initiative. Woohoo! That's awesome. Spending most of her time investigating hellbender health and amphibian conservation, but she also led a lot of our working teams of microbiota, and she also helped organize some graduate student group meetings and also team science events. So really was very active in our initiative and very, very engaged. Her current role focuses on research and response to avian, herp, and aquatic animal diseases within the state of Florida, with research goals to bring veterinary diagnostic and laboratory techniques into field-based wildlife conservation projects. And with that, I will give you Becky.
All right, thanks, Deb. It's great to be back in Tennessee virtually. Yeah, so today I thought I would pick two projects that would be interesting to you all in the One Health world on wildlife diseases here in Florida that focus on bird and reptile populations. So hopefully I know how to use Zoom by now. There we go. So it's really, actually, I love the state of Florida a lot. I love getting back to as close to tropical ecosystems as possible. And it does not disappoint. So it's really, really cool to see the wildlife diversity and the habitat diversity we have in Florida. But with that, we have a lot of human alteration and effects of human populations. So we have a lot of amazing challenges to parallel those amazing ecosystems. So if you're working in wildlife disease or wildlife conservation, it could be frustrating, but it can also be very interesting, some of the challenges you're facing. Oh, and this is just the amount of human wildlife interactions or pet wildlife interactions are pretty amazing here. So here in FWC, we're a state wildlife organization, but what I find really cool about FWC is we have an entire research branch, which is a little bit different than some other agencies, and it's the Fish and Wildlife Research Institute. And then under that, we have a Fish and Wildlife Health. So a really good group of both aquatic health specialists and then what I think of as wildlife health or terrestrial health specialists doing surveillance, population health research, any kind of morbidity or mortality response, and then also providing health technical assistance to anybody else in the state who needs it with respect to wildlife health. And then specifically the Herps and Birds Research Program, that's under me. We are very small, little program, me and a few other people trying to bring anything from the field to the necropsy room and then to the bench top to answer small or larger questions in wildlife disease. We're here at the One Health Initiative today. So most of us here understand the One Health model where, it's really about connecting sectors in health of human health, environmental health, and animal health. But what I like to think about is how the One Health model works is more of a network. So here to the right, it's not a One Health network, it's just a network graph I took from another group. But I feel like it really highlights that when we're working in One Health, you don't necessarily have to have everything all the time, but just understand that if you're working in a project at one of these nodes, that node has several branches. And as long as you're acknowledging those branches and trying to find people on the other side of those branches to help you with your project, you're going to have better outcomes. And because of that, I know this is belaboring probably back to third grade, work in teams, work well together, but it really is important, and it actually is very, very hard. And so collaboration to get to this One Health solution to a lot of these problems is through collaboration and having very, very good teams. And so for the wildlife health program, one thing we've really been pushing is having functional teams. So everything from health, ecology, and lab analytics working together and covering all of the bases of how a team works, and really having that functional, having the team happy, productive, and also creative, so you can really use the best of your abilities from your people. And so hopefully these two projects I'm talking about highlight some of those important aspects of teamwork. So first, I'm going to talk about gopher tortoises. I have always loved gopher tortoises, but never really got to work with them until I came down to Florida. So it's really been exciting to have the ability to kind of get close to these guys and get more into them, because they are super cute and very, very important animals. For all of you who don't know much about gopher tortoises, they are a tortoise, and they're the only tortoise here in the eastern United States. And they are really, really important animals in the ecosystems they live in, which is in the coastal plain of the southeastern United States. I am not a gopher tortoise expert and biologist, but if I could generalize, they like warm, sandy soils in which they can build their burrows and come out and forage and eat the grasses that they need. But because they build these really big burrows, they become these ecosystem engineers and are really, really important for so many other species that use these burrows for part or maybe all of their life cycle, including the indigo snake. So that's kind of that guy kind of right up here. And you can see that gopher tortoises historically are actually throughout most of Florida, panhandle down to peninsular Florida. Yet they're having, and because of that, are facing a lot of threats. So I came onto the gopher tortoise program because there is this growing need for better understanding gopher tortoise health, not just whether or not the population is healthy and whether or not the habitat is healthy, which there's a lot of work being done on that. And it's really, we're really learning a lot, but just understanding individual health parameters and what that means for population health and how we can use that to help us manage populations. And specifically, there are a lot of translocations happening with gopher tortoises throughout the state due to permitting where there might be a development or some issue where tortoises need to have to be moved and then transferred somewhere else. And there's still a giant gap in what pathogens might be transferred, whether or not that matters, and whether or not an animal is sick. How do I identify whether or not it's sick, and whether or not these animals survive due to all of these parameters? On top of that, there are a lot of people noticing mortality and morbidity events with gopher tortoises, and there are still a lot of people doing great work with health in gopher tortoises, but it's such a large need that there's a large gap still. And so me being a wildlife veterinarian down here, hoping to jump into this and help out figure some of these issues out. And one background information I want to say about gopher tortoises and the gopher tortoise health world, there is a good body of literature looking at what's called upper respiratory disease in gopher tortoises, or URTD. And this has largely been attributed to the bacteria Mycoplasma, but now the genus is Mycoplasmopsis. A lot of work has been done in the counterpart in the western United States, the desert tortoise, where these pathogens may have been introduced or at least causing issues with translocation or wild populations, actually causing significant effects on population status and viability. Here in the eastern United States in gopher tortoises, it's a little less clear about whether or not Mycoplasma was an introduced pathogen and what kind of problems it's causing now. But a lot of focus may be just towards this Mycoplasma, with a lot less focus on some of these other pathogens that not only can cause upper respiratory signs, but maybe just cause disease in gopher tortoises that we don't want to overlook. And so to start with any pathogen, you always want to make sure that you're getting the good baseline data you can use to compare to know what is normal and what is abnormal. And so when we're, what I wanted to go into with this project is kind of a two-tiered approach. Is one is, how are we going to evaluate the cause of these mortalities? Unfortunately, a lot of the mortality events that are being documented end up being these dried shells, and people want to know why they died. And unfortunately, if you don't have a body, you can't figure out why that animal died. Not that shells aren't useful, but it's really just kind of the tip of the iceberg on figuring out what's happening. And so really, really starting to push that if we get a fresh carcass, we really need to get that immediately processed, especially for histopathology. And then also, make sure that we're recording as much as possible for this baseline data, so we know, what is a normal pathogen to find out there and not automatically attribute to that, to the death. And the reason being is I have an example here of a newt mortality event in Tennessee in which there was a mortality event. Initially, we thought maybe it was this chytrid fungus, if you know much about it. So we went to the pond to figure out what was going on. We did swab these animals for chytrid, but the good news is we did not stop there. We actually removed sick and dying animals for full histopathological analysis. And what's interesting is that several animals were positive for this chytrid fungus we were worried about, BD specifically, not B-Sal for folks who know the difference. But histopathology actually showed that these animals had really severe coccidiosis, something we'd never even thought to test for. And because of that baseline data, we were able to actually understand that this event was a coccidiosis-driven event, and BD was actually just present, and it wasn't actually causing disease. The second approach to this Go for Tortoise plan was not only take these cases and understand why an individual died, but making sure that we have an actual sampling design that we can answer subtle effects of pathogens on these populations, which requires a lot of work in sampling populations, specifically in certain areas with certain factors, a large data set that you can actually analyze appropriately and look for subtle factors. And again, another reason for this is a lot of questions are there sublethal effects of pathogens? And it's really, really hard to say about sublethal effects unless you have a large data set. And one example, again, this is a project with hellbenders. As you know, in Tennessee, we have a lot of them, or used to. And so I was sampling for pathogens with them. We did get a few that were positive for this ranavirus, but really didn't know what it meant. But over time, we were able to get enough samples to actually see a trend that was statistically significant, that animals that were positive for ranavirus on sampling had lower body condition than those that were negative, showing that there probably are sublethal effects of ranavirus potentially affecting survival and fecundity in these animals. We don't know that, but at least that's a start. So with all of these plans, we wanted a sampling strategy, and to get all of these data, we need partners. And really what it is these are already people, biologists and rehabbers, doing all the groundwork, and then I am partnering with them to help them add on a layer of health. And so this is with a lot of different groups. Tall Timbers Research Station has been a main player in this, Archbold Biological Station, and two main rehabbers have been Swamp Girl Adventures and the Florida Wildlife Hospital. But then also a lot of FWC biologists and many others working together. So for these, we're devising a sample collection, so ante-mortem at the time of live sampling. Tall Timbers specifically has started a very, very large-scale program of getting health samples on all of their gopher tortoises that they're sampling. And then this is Deanna Riente, who's actually making it a master's project of looking at individual health and pathogen status over time, getting choanal and colloquial swabs for PCR, and then also drawing blood and doing a lot of extra diagnostics, evaluating health from blood smears, whole blood for PCR, serology, and then in some animals, fecals as well. Post-mortem, we can still do necropsies. We do them opportunistically and as quickly as possible for fresh tissues. Again, we're doing swabs as well as testing tissues instead of swabs for certain pathogens to get better detection. And what's really been a great partner is the Wildlife Epidemiology Lab in Illinois, headed by Matt Allender, also used to be from UT, and Laura Adamovich, researcher over there. And they have devised a fluidine chip assay where they can do qPCR assays for all of these pathogens of interest in one fell swoop, which makes it really a lot more comprehensive than just pigeonholing yourself on one particular pathogen. What's really awesome too, because you can add so many pathogens on this fluidine chip, they're able to add maybe pathogens we wouldn't think gopher tortoises would have, but that might have a human health component and might be in a reptile. So things like Salmonella, Leptospira, and some other just pathogens we just know very little about in wild reptile populations. So we did that. We collected swabs from rehabs as well as the fields. And we're starting to gather a lot of these data, send them to the wildlife ecology lab, as well as doing some in-house PCR for some, small ranavirus cases and accumulating those data. And so from all of that, these are detections so far. So we've gathered almost over 200 samples. We actually have more that are still in analysis right now, but these are the 5 top pathogens that we're actually seeing in gopher tortoises from the field and from the rehab. And if you look here, the bar plot tells you how many animals were sampled. The gray is negative animals and the brown is positive animals. And the big ones here I'd like to mention are this Helicobacter species, which I will talk about. We are finding human pathogenic leptospira in gopher tortoises, is really interesting. We're finding mycoplasma still, but that's not really unexpected. We're getting ranavirus from rehab-only animals and 1 field animal. And then another unexpected is this fungus Aminomyces. And again, something we didn't really think to test for, but because it was on the Fluidine chip, we got data for it. And then from necropsy cases, what was really, really interesting is not only were we getting swab data from these guys, but like I mentioned, we got histopathology and gross lesion data. And we actually had 5 clinical ranavirosis animals. So not only were they positive for ranavirus, but actually showed clinical disease and likely died from ranavirus infection. And this is one of those cases where you can see hemorrhage throughout the GI tract. And then this animal actually had a blood smear with it too. And this is an actual inclusion for ranavirus in the blood smear. And because we had all of these ancillary diagnostics, we could really say with certainty this animal died from ranavirus. So I'm going to go through some of the pathogens and their implications. Leptospira was a huge surprise for us, but after talking to other states who are also using Matt Allender's lab, they were also surprisingly getting some positives. But we really don't know what this means. It's a positive on a swab, and we got about 20, or 5% of these animals positive. But what was cool is Tall Timbers was also collecting blood and banking plasma. So they were able to then submit for serology their plasma samples to a lab at the University of Illinois with the help of this lab. And actually 72% were seropositive, meaning 72% of their animals, regardless of their qPCR status, had evidence of exposure and immune response to the Leptospira pathogen. So they're actually showing now that we have exposure and several serovars. For people who know much about lepto, a serovar might tell you where this Leptospira particular where they got it from, but it's still really unclear because we have over 5 serovars documented in these guys. And I think it's a really, it really needs to be investigated further because Leptospira is a very important pathogen for humans and domestic animals. And whether or not gopher tortoises are also being affected or potential carriers is still unknown. Like I said, ranavirus is another one I think is really important that we gathered these data for. It was only detected in carcasses, so it might be that, when we're not going to find ranavirus in a live animal because potentially most gopher tortoises, at least in Florida, are dying from it. I know there's other cases in which gopher tortoises have been found positive without clinical signs at the time of testing. But what was really interesting to me is that we actually have cases showing ranavirosis, which at least in the published literature has not been found before. And whether or not that means this is emerging, we still have no clue, but it's, I think, an important baseline. Helicobacter is another one. This bacteria can be found in normal stomachs. And there's different species of Helicobacter in different host animals. However, in gopher tortoises, there are studies showing that there is severe disease associated with Helicobacter in a few cases. So again, this brings up the question of whether or not Helicobacter is potentially just there and we're picking it up. What I'm most interested in is potentially, Helicobacter might be in the stomach, but if you're picking it up from an oral swab, potentially that's a prognostic indicator for this animal, saying that it's sicker than normal and potentially an overgrowth of Helicobacter is being detected. Still, we have no idea, but now it's something we need to focus our attention to. And lastly, I won't harp on Aminomyces very, very much, but this is an emerging pathogen known in freshwater turtles. It is a fungus causing shell disease. Why we're picking up in choanocloacal swabs in gopher tortoises is a little bit of a mystery. Potentially some of the fungal DNA gets on the swab, but now we're actually starting to swab shells as well. And the reason is we don't think this is incidental in some of the cases, because one of the animals that were positive from rehab, this is a picture of that animal that was already thought to have a shell disease even before we knew it was Aminomyces positive. Still, we don't know if it's causative, but it's something that we need to investigate further. So in summary, all these pathogens that we did not know a lot about, we still don't know a lot about, but at least we have a little bit more baseline data. And what I'm really excited about, especially with the project going on at Tall Timbers, is they are now going to pair these pathogen status data with actual behavior and actual clinical signs and survival of these tortoises out in the wild to see if there are actual health implications for these. This is everyone to acknowledge just a whole bunch of people. And I don't know, I have a two-part talk. I could take questions now for Go for Tortoises, or I could just keep going. I'll let Alyssa and Ashley decide what's better.
I think you can keep going. I think it's good.
All right, cool. All right, so we're going to just totally switch gears, and it's kind of more reality anyways for anyone who works with multiple projects, and move straight on to black skimmers. This is also a really interesting project where as soon as I started, I was approached that they had an issue with septic arthritis in black skimmers, and they wanted to evaluate it further. And they being a lot of FWC and Audubon biologists. So what was going on for 2 years in a row, 2 different black skimmer colonies were experiencing actually pretty high mortality rates in their chicks due to the septic arthritis. And if you look, this is a picture here of one of the chicks, and you can see a swollen joint. And these animals were falling over, not doing well, and eventually dying. And this was specific in 2 colonies in Southwest Florida, near Marco Island and Fort Myers Beach, and it happened 2 years in a row. So now this red flags, you know, what's happening? And it's associated with Staphylococcus. And the going thought was that they were also seeing a lot of sand spurs on these chicks, potentially the sand spurs introducing a bacteria or poking them. And so people just wanted to take a breath and do a better evaluation. Now, there was already great work already being done out of Squidus, which is the laboratory out of University of Georgia, led by Nicole Nemeth, looking at the histopathological findings of these carcasses and really characterizing that, yes, these chicks were dying from a septic arthritis associated with a Staphylococcus, which they were able to culture. But is that it? Is that just the end of the story? Or, you know, our biggest question is why? And the reason is the sand spur plant is a native plant. So why are these chicks all of a sudden getting all of these sand spurs, getting poked, and then dying? And so potentially, is there an increase in sand spur plants due to things like climate change or habitat alteration? Are the chicks just less aware of the sand spurs? Are they immunosuppressed from something else? Is there something we're missing that's actually causing them to be more susceptible to staph septicemia? Or is there something changing in the Staphylococcus specifically? Is a natural staph becoming more virulent? Or what a lot of people were worried about, is this staph not naturally occurring on their skin? Is it actually coming from somewhere else, and are they picking it up? And for those that don't know much about Staph aureus, you know, people have a Staph aureus strain on their skin. There's Staph aureus in other bird species like chicken, notice, and it's usually a normal flora that becomes a problem when you have a disruption in the skin barrier. So we're trying to think of all these reasons why all of a sudden these chicks would get this from a native. So we formed a team, and we had a lot of questions. And the ones that are grayed out are questions we are answering, but in a different capacity where someone else is leading that. But our top 3 questions are, you know, first of all, let's figure out, are there physiological differences in the chicks between these colonies, you know, between affected and unaffected sites, to answer that question of potential immunosuppression. But then also, you know, do we want to know about the staph specifically and potentially other microbial organisms that might be playing a role? So are there changes in the microbial assemblages in the environment and the skin of these chicks at these colonies? And then specifically, let's look for staph in all of these samples and see exactly where it's found. These other 3 questions are ongoing. And one of them that we're really excited to get more information on in our collaborating lab at Clemson, should be finished soon, is figuring out what these Staph aureus isolates actually look like. We did test for harmful algal blooms, just FYI, I'm not going to talk about them, but we didn't get any hits really. And then there's a whole side work looking at the sandspur plant in different conditions that might make it grow quicker. So this was a project. It was a 2-year project, getting as many samples as possible and then processing them to answer those top 3 questions. We're collecting water, and then we're also doing live sampling of these chicks during nesting season for both blood and swab. All right, keep going. All right. So this was 3 sites over 2 years. And to the right, you can see the map. We have Carlos Beach and Marco Pass. Those are our 2 affected sites. And then we added in a skimmer colony we were able to sample as well, Lido Key in Sarasota County. And so we did 2 sampling events at each site, 1 each summer. And then for actual environmental samples, we're able to go out 3 times a year to capture seasonal variation in those environmental samples. And this was actually all done by Michelle. She went out, rain or shine, when she had a date specified for environmental sampling, she was out collecting. And this was even very, very close after Hurricane Idalia. So she really, really sacrificed a lot to get these really important samples for us. And these are just some pictures of live animal sampling. It's actually extremely complicated. And again, we're talking about teams, required a very comprehensive team, all the way from people who are able to handle birds, people able to draw blood, people able to set up a sampling event at night on these beaches, with everything, working together for animal health and human health and actually getting quality samples. Like I mentioned, from these birds, this is just kind of sample breakdown of what we're able to collect. So this is a skimmer chick here. On the left is all of the blood samples we're able to get. So we did a blood draw with the help of both Save Our Seabirds veterinarians and Crow, and getting both plasma, protein, electrophoresis analysis, chemistries. We did a total solids in the fields. We did a PCV in the fields, which is percent cell volume, and then actually did blood smears as well. And then we got a lot of health data, both body condition and a physical exam. We quantified sand spurs on these animals, took swabs of the leg, regardless if they had a joint lesion, and then from those swabs, extracted DNA for microbial sampling. And these are just kind of breakdowns of the samples we got. So we ended up getting 45 each of the environmental samples, and we were able actually to get a good amount of sampling from live chicks. But you'll see here, the only issue is Marco Island. We were not able to sample in 2023 because the colony did fail. We had a predation issue and they didn't nest. And so that colony just went away. And so we really only had Carlos Beach and Lido Key for each year. We ended up getting about 57 chicks total. And then processing samples in-house, all of our environmental samples came through here at the lab. This is Emmett, who actually spearheaded how to get a good DNA extraction out of a 50 mil sand sample. And we actually experimented for several months to figure out the best way. And it's pretty exciting, using what we call the slurry method, which we had help from other researchers giving recommendations to get a really good DNA representation of that sand community. Extracted from the swabs as well, and then actually sent them back on over to University of Tennessee to the sequencing lab there with Veronica Brown to get our sequence outputs. And then blood samples, same thing, all the plasma and blood smears. We sent them off to the University of Miami to be evaluated, since they do really well with non-traditional animal blood samples. And for our data analysis, we looked at both the community of white blood cells and plasma protein electrophoresis, the plasma fractions, as well as the microbial communities, and all of these samples associated with these 3 tiers of predictor variables. So we're looking at site and year, what kind of site it was and what year it was. And then for the swabs, for the individual data, swabs and blood, we were able to look at individual variables, their body condition, did they have joint lesions, were they sick, did they end up dying, how big they were, and whether or not they had sand spurs. And then at the bottom here, for specifically for microbial communities, we really wanted to look at, do these microbial communities change based on site and year and all these factors, but do they also change by sample type, sand versus water versus swamp. This is Andrea Silva, statistician here at FWC. We worked very close together, and she did an amazing job of really helping us do the best with these analyses. So the first question we want to know is, are there physiological differences in these chicks? And this is really going to be through the blood data. And what we found is, after throwing these in these models, there was an increase in heterophils, and heterophils are a white blood cell associated a lot with bacterial response, not all the time, also stress, in animals that had swollen joints and sand spurs. So you're thinking, okay, well, these animals had swollen joints, they had sand spurs on them, and then they got bacteria, and now they have increased heterophils. And that makes a lot of sense, and maybe, it's a no-brainer, but it's really nice to actually see that pattern and confirm that that potentially is what is happening. Protein fractions, we actually had we had significant differences in some of the protein fractions as well, specifically albumin and alpha-1. And I am still learning, the significance of some of these fractions in avian species. Please feel free to talk to me afterwards if you have any suggestions. But at least we're seeing some differences between animals. Again, the only ones that came up as significant were ones with or without sand spurs on them and with or without swollen joints, showing again that there is potentially an inflammatory response to sand spurs and then resulting in swollen joints. The blood chemistry, the analysis was done a little bit differently. It had to be per chemical analyte. And we actually did get some interesting results. We're still kind of sussing out. This data set was a little bit smaller than some of the other ones. So, but still, these are some values that were statistically associated with these factors. The first one is site and year had a very strong change in AG ratio. So that's kind of interesting, especially in the sites 2022 and 2023 in the affected sites, which is kind of interesting. We'll talk about that a little later. Calcium apparently had an increased trend in animals with increased spurs on them. And then the liver enzyme AST had a very strange association with whether or not an animal was anemic or had too many red blood cells, which might mean they're a little dehydrated. So these are all factors we are still sussing out. But it's good that we have these data. It's data never really had this comprehensive before in a wild species like this. And the big question, though, is, are there changes in microbial assemblages in the environment and the skin? This is one of our really big questions we want to answer. And so first off, just looking at the microbial assemblage data based on sample type. So different colors represent different top genera from our sample. So you can look at the top here. These are sand samples based on sites. Big Marco Pass and Carlos Beach are on the left. Those are our sites affected. And then Lido Key is our site that's unaffected. And if you look right away, sand across sites, there might be some differences. So each one of these bars represents one sand sample from that site. But what I'd like to show more is that across sample types, there actually are some big differences. So sand just looks different than water, and it looks different than swab. And specifically in swab, what I'd like to show is that this pink area, which you cannot see on the key, but I will show later, this pink is Staphylococcus, and that sequence specifically represents Staph aureus. And you'll notice there's a lot of that in the Big Marco Pass, in the Carlos Beach samples, less in the Lido Key, and almost absent in our environmental samples, really showing that the Staph aureus might be from the skin. When we're looking at just the differences in the microbial assemblages based on these different factors on these swabs from these animals, we did find that microbial assemblages did shift significantly based on whether or not an animal died, whether or not it had swollen joints or looked sick, and whether or not it was anemic or not. So this is really cool data to show that microbial communities on the skin are shifting and associated with these health data. Now, whether or not they're causative or not, and they're probably driven by Staph aureus, is another question, but at least we're showing that there are changes that we should be aware of. So we did a bacterial species analysis, which basically looks at the top bacterial species and which ones are significantly associated with a sample type. And what we found is time and time again, Staphylococcus was significantly the top indicator of the following groups. It was the top indicator for a swab sample over environment. It was a top indicator for samples, swab samples from affected sites, swab samples from year 1 or year 2022, and from individuals that had sand spurs, were sick, or had swollen joints. So again, we're kind of painting this picture of Staph aureus on the skin is actually associated with all of these things. So our last question is, where is Staph aureus found specifically? We just hone in just on that particular bacterial species, and you'll find that the relative abundance from these samples is significantly associated with site, and it's significantly associated with year. So this is relative abundance out of 100, and you'll notice that treatment sites, so sites that are affected by this disease, have higher Staph aureus loads on their skin, and they're higher in 2022. This is another bar plot showing Staph aureus across all samples, and pink is Staph aureus, green is just any other bacterial species. And you'll notice that sand samples don't have it except for this sample here, and water samples also don't have it except for just a little bit in this sample here. However, lots of pink on the swab. And what's really interesting is, so these swabs are from Big Marco Pass. That's from year 1. Animals had that disease in year 1. We didn't get Big Marco Pass in year 2. These are samples from Carlos Beach. The first half of these samples are from year 1. So you'll notice year 1 had, some of them almost had 100% of their bacterial community on that swab was Staph aureus, whereas year 2, they really, really dropped down. Lido Key across all years, this is our reference site, had Staph aureus on their skin, but never really what we consider an overgrowth of it. So one thing I wanted to mention, you might have noticed there was an effect of year. So we noticed that 2022, we had a lot more of Staph aureus and these shifts, whereas 2023, we didn't. There are 3 factors that actually could explain this that really help us understand this disease a little better. Is first of all, we didn't see actual joint lesions in 2023 in the Carlos Beach area. So we didn't have this event happen. And 2 things that might be a player is that in 2023, biologists went in and chemically managed for the sand spur plants. At Carlos Beach, what has been going on is there's grooming for sand spur plants to remove it from the beach each week because no one wants to step on it, right? Then we have these issues. However, as soon as the black skimmers colonize the area, you have to rope the area off, and then you can't groom for it anymore. And the problem is when you groom for these plants, a lot of the seeds are released, and the plant can actually grow back a lot quicker. If you groom every week, it's not a big deal. But once you rope that off for the black skimmer colonies, it was amazing to see how dense these sand spur plants were. And so they actually went in 2023 and chemically managed for them inside that roped area. And lo and behold, we lost that Staph aureus on the skin, and we lost joint lesions. So in summary, no big surprise, a lot of work went into this for this kind of summary, but it's still really important, is that this lesion, this disease is highly associated with the sand spur plant and likely the punctures and letting Staph aureus enter and take over in a disrupted skin environment. The microbiome and blood profiles really help support this. And that what's really important, it's likely not a bacterial contaminant in these communities. But there's still a lot more studies underway. And really what I'd like to hone in is environmental management is key. If you can't have a natural ecosystem, you might have to go in secondarily and be creative on how you're going to manage these environments for these animals that don't have a lot of choices anymore on where to put their colonies. And so, they may be limited to these less than ideal areas, or potentially have to deal with sand spurs that they wouldn't have to deal with before. So, and one health plug, there actually are a lot of studies I don't know a lot about, but I'm getting familiar with. In a lot of human cases in Staph aureus infections, in crowded environments, in coastal areas that might actually overlap in the pathophysiology we're seeing here with black skimmers. And finally, this is just a video of a juvenile skimmer that's just really, really cute. They're they're much easier to catch than I thought because they just kind of sit there. But there he is, just looking for somebody to help. And acknowledgements for this was also a very large team, everyone from biologists doing banding, people in the lab, everywhere in between. It required so many people to coordinate, and I'm very grateful for that. So with that, I think I will take any questions.
Awesome. Thank you, Becky. Those are awesome case examples and two really different but really interesting sort of things that you've been looking at. So does anybody in the audience have any questions? If you don't feel like unmuting, you can post them in the chat.
I have a quick question.
Yeah.
Did you guys look at all if there was any association with the number of puncture wounds and the development of disease, if they were like stepping on more than others?
Yeah, so for this 2-year study, we didn't quantify puncture wounds. Most of this was from live animals. I do have some necropsy data where we have those data. And then in the 2 years before me, all that necropsy data from Nicole Nemeth, they do a very good job of actually quantifying punctures, and they do see a lot of them. We did quantify sand spurs, so we did count the sand spurs, and there are data showing maybe a correlation between sand spur load and some of these values, but not the wounds themselves.
Thank you.
Anybody else? I have a question, Becky. I actually have two questions. One, because I'm interested in wildlife toxicology and pesticides and herbicides, I'm curious, do you think that the sand spurs have become an issue because they're overgrown, because other competitive species have been sort of taken down, or like I'm wondering if there's like a potentially biological mechanism that we could control the sand spur plants?
Yeah. So what I'm about to tell you is all anecdotal. Okay. So I'm still learning about the ecology of these sand dune ecosystems. I'm learning slowly, but some interesting anecdotes of the sand spur is in the Carlos Beach area specifically. Yet you're right, because no one likes them, it's become a little bit artificial, and there's a lot of that grooming. And I think the grooming itself is giving the sand spur an advantage if you don't continue to groom, because you're just ripping everything up, and then they're very, very quick colonizers. And once you reseed and strip the land, these guys come back with a vengeance. And so that's what's happening because you have to rope it off. You can't groom a colony because you'll just run over their eggs, as you know. So that's been an issue there. Apparently with the hurricane, there was some shift in the dunes a little bit, and people couldn't go out and groom. And there did seem to be some reduction in sand spur growth naturally. So anecdotally, could we create natural dune systems to solve this problem? But, it's really tricky because, we have this beach, you have people trying to access the beach. So it's understandable they don't want to stand on sand spurs. So it just, at this point in time, the Marco Island population is a protected area. So that one's been a little bit more of a mystery of why the sand spur would be overgrown. And so there are trials looking at climate change and things like that, maybe. Yeah.
Okay, great. My other question was about the fluid chip, the fluid diagnostic chip.
Oh,
I have never used one of those, and I just wondered, is it like several ELISA tests just on the same?
Yeah, no, it's really cool. It's using that, I guess, a flow technology through a chip, but the same technology for any quantitative PCR. So the cool thing is you can load all of your primer and probes for each qPCR assay onto that chip, and then it runs them all at once separately. So it keeps the costs down considerably, as opposed to you having to run all those different assays separately. Yeah, so it's all qPCR.
Yeah. Okay, sorry, I missed that. Thank you so much. That's awesome.
Yeah.
We've got quite a few more minutes if anybody else has any questions. don't see any have popped up in the chat either. Okay, well, I'm sure Dr. Hardman would be happy to share her email if you guys have any questions. Otherwise, you can let us know, and we can forward those on. But just again, a reminder, if y'all check the chat, those links are there if you're interested in being updated when our kayak river cleanup becomes scheduled. Feel free to put your contact information in there. And we'll see you guys back the last Thursday of April for our next One Health Luncheon seminar. So thank you all for joining. And Becky, thanks for being here. We really appreciate you sharing these cases with us. I thought it was very, very interesting and makes me want to get out in the field and find some other mysteries.
More questions. Yeah.
Absolutely. Thanks, everyone.
Right. Thank you.